Okay. So, that's the fun stuff. Okay. That's the fun stuff. So let's finish up here. The last thing we got for today, guys, is going to be this thing, prokaryotic cell division. All right. When it comes to it, I don't really have, well, we're going to talk about it. It's just binary fission. But I'm not going to get too hardcore. I'm not going to ask a short answer exam question on it. I feel understanding what's going on inside our bodies is a little bit more important. So that eukaryotic mitosis is what I'm probably going to ask. I could ask you to compare the two. Okay. Prokaryotic cell division and eukaryotic cell division. I could do that. I could just ask it as a multiple choice question as well. So, I mean, I've done it both ways. I'm not going to have like 15 short answer questions, obviously. I may just have 2 or 3. But if you can answer it as a short answer question, guys, you can answer it as any type of question. All right. So without further ado, here we go. Prokaryotic. What is this? This is bacteria, right? When we look at these guys, prokaryotes, because let's see here, what do we have? I believe in, I can't remember, 12 maybe? In chapter 12, I believe. We'll look at how the DNA is read, how it is truly copied, and how we truly translate it. I believe is chapter 12. So we'll look at prokaryotic, the how, a little bit later. We'll look at eukaryotic and the how we actually read the DNA and copy it and make stuff out of it. So, but just briefly describing it though, I mean, prokaryotic genetic material. Oh my God. It's like super simple and super complex. It's just, there you go. Ta-da, there it is. Just a circular ring. That's it. Just a circular ring. That is like insanely simple compared to ours. I have some examples of how fast I believe it goes on maybe the next slide. But much more easy, much more simpler, and a hell of a lot more faster than in a eukaryotic organism. So again, remember they don't have a true nucleus. Remember, it's just a nucleoid region, where if I have, this is my eukaryotic cell, there's my nucleus, and then it has the DNA inside. When I look at a bacteria, a prokaryote, okay, they just have DNA kind of in the middle there. There's no double membrane around. There's no nuclear membrane around it. So hopefully you remember that. So because it is in this kind of circular pattern there, and there's no kind of plasma membrane around it, I mean, all it simply does is it just reads it, copies it, and boom, I got 2 circles now floating around. So prokaryotic cell division is a hell of a lot easier. There's not all the hubbub and separation and moving it out. It's just DNA duplicates and then the cell divides into 2. That's it. Pretty easy. So what we call prokaryotic cell division is binary fission. Okay. So if I asked you, what is replication of a bacteria called? Fission, binary fission. Okay. One cell just divides and becomes 2. That's it. Okay. That's it. This is referred to as asexual reproduction. Okay. This is referred to as asexual reproduction. Okay. Mitosis is asexual as well. Okay. So I don't really understand the whole hubbub of putting it here and not putting it in with with mitosis. Whatever. Binary fission. It's non-sexual. But this is also non-sexual. So I don't really get the asexual part being talked about only here. Mitosis is asexual as well. When it comes to a eukaryotic cell dividing, when it comes to a prokaryotic cell dividing, prokaryotic cells divide like every 15, 20 minutes. Some maybe go a little bit longer, some maybe a little bit less, but each new generation pops up every 15 to 20 minutes. No wonder sore throat. if you don't get on top of it with medication, how it can just explode and you can have a massive infection. Because every 15 minutes, that sucker is dividing. And there's more and more of the bacteria causing all that sore throat. I believe it's around an hour-ish time for eukaryotic cells to copy the DNA and then divide. So obviously, prokaryotic binary fission happens a heck of a lot faster than a eukaryotic cell. And, both give me genetically identical. Bacteria becomes bacteria. Skin becomes skin. Liver becomes liver. Yada, 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 yada. Okay. So when it comes to a function of fission, it's basically there to increase the numbers. Okay. If they die, they die. Okay. Ivan Drago, right? If he dies, he dies. This is not really there to, perpetuate really any survival. It's just there's just rapid reproduction. And the ones that survive, well, they have a potential advantage and they can potentially pass those on to the next generation. Okay. So this is producing more and more. When it comes to multicellular guys, fungus, plants, animals, okay, this is growth. This is renewal. This is repair. So they're not necessarily, obviously cells are a little bit different in us than in a bacteria because that's all they are, is a single cell, where we are a collection of billions of them. So for us, it doesn't really help us to magically become more of me or more of you. It's just growth and repair, renewal. Where in prokaryotes, it's given rise to the next generation. Okay. And for us, that's what meiosis is for. Yeah. I mean, there it is. Go back. I'll come back to that video here. But there's the circular DNA and oh snap, it's copying. See, one's red and one's purple. Okay. Oh, it's elongating and it's dividing and then ta-da, there you go. This is super basic and super simple. You can see why I wouldn't ask a question, right? Because it's so easy. It's too easy to ask a question on. See, asexual. Okay. Growth and repair. They don't really have cytokinesis associated with it. It just happens. Okay. It just pinches in and divides. There's no doubling up. It's already doubled up. So pretty basic. Pretty basic. So yeah.
Bacterial cells increase in numbers by the process of binary fission. The parent cell grows in size while doubling all of its contents. Then it divides to become two daughter cells. Each daughter cell is identical to the parent cell. Two cells become four, which become eight, and so on. This results in exponential growth of the population. The time required for the number of cells in the population to double is called the generation time. Depending on the medium, temperature, and other conditions, some bacteria can double in minutes. Other bacteria have generation times of several hours or days.
Like we care about armadillos, right? Good Lord. Sometimes the stuff these people include in, who cares? All right, this is another cool one. This looks at this concept of bacteria surviving and advance. Okay, they survive the application of this antibacterial medication, So now those that survived were exposed. So now next time they're exposed to it, they're like, I got the goods to beat this. So I'm going to survive. And then, so we have to come up with new medication and then we kill it all off. That's why, following the doctor's orders and consuming that, you stop taking an antibiotic when you feel good, right? Not generally when you go through all of your medication. But by not doing that, we've allowed some of these bacteria to survive, and now they have a ability to survive the application of that next time, just by these random mutations that occur when they read their DNA. So we have all these superbugs out there, right? Now we have some that aren't. You can't kill them with any type of antibacterial. So here's a video that kind of shows that off here. Let's see if it works.
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands. And at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.
Isn't that just crazy? That's crazy. That shows you how fast. That shows you, man, just how fast bacteria can respond, how fast they can evolve. And we'll talk about the process of being able to survive and advance evolution. Okay. We'll talk about that's in the last unit here. So, this just kind of gives you kind of a little bitty of a precursor to some evolutionary things that can occur in a very simple, very basic single-celled organism here. When it comes to us, eukaryotic organisms, animals, plants, it's a little bit more complex, obviously. But I mean, this is the world we live in now, right? Where you got all these super bugs, and you could just see, the totality of your health and what it actually means for you in terms of your ability to fight crap off. When it comes to COVID, there's been other COVID species. We've been able to handle them though pretty decently. This one is very different because where it attacks the cells in the lungs that code for certain things, and we have an inability to turn off those things, and you get this overproduction of activity in there, and you mount an immune response, and the body just goes in there and it sees the foreign invader, and it can't delineate at the time who's the good and who's the evil. So it just, you have this massive immune response in your lungs, and that's never a good place for a massive all out war to occur in the body. And you get accumulation of fluids within the lungs. And, if you have inflammation already going on in the body, diabetes, obesity, any type of pulmonary type thing, asthma, okay? If you have all these comorbidities, your ability to fight this thing off is dramatically less than if you didn't have those comorbidities. So, your health up until this point, do you eat well? Do you exercise? Do you get to sleep? I mean, is it awesome and fun to go to bed every night at, 9 o'clock? Not necessarily, but I mean, I have a family history of cancer, so I don't have a good potential background. My mom's mom, she had breast cancer. She beat that. My mom's dad had Alzheimer's. That sucked. My dad's mom had type 2 diabetes. My dad smoked. My dad died at the age of 43 from a heart attack. He smoked. He didn't eat well. My mom passed away a few years back from ovarian cancer. She beat it once, couldn't beat it again. But it's the totality of health and the choices that you make, and clean diet, exercise, psychological aspect is, there as well. But nutrition and understanding, some of these basic concepts when it comes to how bacteria divide and what it is and how they get in the body and how they interact with it. That's some of the most important things that you can know. So for me, that's why, the talk about cancer, I enjoy talking about it. I don't like what it did to anything in my family. But, if I can impart any wisdom, so you don't have to do that, and damn it, then something got accomplished and all that nasty ass stuff. You guys have any questions about anything? You've already done your due diligence for the day over here by answering the question. And if you didn't answer the question, put your name over there. I don't know why you didn't answer the question. Didn't want to participate. That's fine. Get that redemption exam 2 done. Okay. That way you can move on and start worrying about 9, 10, 11, 12, 14. Okay. For the next exam. All right. If you don't have any questions, I'll see you Wednesday. We'll talk about meiosis.